If there's no treatment left, you need to make them. Sijbrandij, GitLab's co-founder, said that while describing what he did after osteosarcoma came back and standard options ran out. This story comes from the OpenAI podcast, where Sid and geneticist Jacob Stern explain how they used AI to chew through insane amounts of biology, and how that turned zero options into a ladder of real ones. It starts like a lot of medical stories start, with a sensation that's easy to rationalize away. Sid was bench pressing, felt pain near his heart, and assumed it was like previous episodes. Except this time, it didn't go away. 2 weeks later, he's in the ER. They tell him, basically, no such thing as a 2-week heart attack. X-ray, sent home. A few hours later, his GP calls and asks, "Do you know how to meditate?" The GP is worried about blood pressure and an aneurysm. Sid rushes back. Good news, the aorta is fine. Bad news, there's a 6-centimeter tumor growing from his vertebrae. Everything after that is urgent and brutal. Surgery to remove it, a frame put in, spine fusion, radiation, and extensive chemo. Sid describes it as medieval, and it sounds like it. And then the part people don't want to hear is that even after all that, cancer can come back. 2 years later, there's remission, then local progression. And for a rare cancer like osteosarcoma, "go look for trials" can translate to "there are no trials." Sid says his oncologist couldn't recommend anything he believed would make a difference. So the project became survival. He quit his day job and went founder mode on cancer. A normal medical instinct is, only order diagnostics if you already know what you'll do with the results. Sid and his team did the opposite. They did everything they could get their hands on, in parallel, because they were out of time. They built what they call maximal diagnostics and put the data out publicly, about 25 terabytes on osteosarc.com. The twist is that maximal diagnostics became a steering wheel. It let them choose targets, choose scans, choose therapies, and sometimes invent therapies. One specific result kicked open the door. Single-cell sequencing is one of the centerpieces. Instead of averaging signals across a blob of tumor, you sample thousands of individual cells and ask what each one is doing. In Sid's case, that helped isolate the cancer cells and spot something actionable. They saw a lot of FAP expression, tied to fibrous tissue. That pointed them to an experimental therapy in Germany, where FAP is combined with radioactive substances. Sid went there twice, then had to stay in isolation afterward because, as he put it, you kind of glow up. It worked. The 2 treatments led to 60% necrosis and about 20% shrinkage. The cancer detached from his dura, and surgeons could go in and scoop it out. They also had evidence suggesting the immune system was getting activated. TCR sequencing showed angry T cells. The suspicion is the tumor microenvironment was suppressive until the FAP targeting approach changed signaling enough to turn a cold tumor hot. They're careful about it. They don't know for certain, but it's their working theory. Then they hit another reality of modern medicine. Even if a target is screamingly obvious, the drug you want may simply not exist in a usable way anymore. Sid's tumor showed extremely high MDM2 expression, off the scale compared to others in their comparison. So they asked the obvious question, are there drugs for that? There were, but development got stopped because, in Sid's telling, drugs reach market when they're expected to be blockbusters. MDM2 wasn't that kind of story. Programs were shelved. So Sid started looking for a pathway to bring the medicine to market anyway. Because even if it helps 10 or 20% of patients, that's still a big deal. This is where Jacob Stern enters. He's a geneticist, not a clinician. But Jacob's real contribution is acting like connective tissue between a ridiculous number of technical domains: single cell, bulk DNA and RNA sequencing, targeted radiodiagnostics, pathology staining, organoids to test drugs on Sid's cancer. And this is where AI becomes less of a buzzword and more like a forklift. One of Jacob's early experiments was blunt. Take a bulk RNA sequencing output, a CSV of genes and counts, feed it into ChatGPT, and ask what it thinks. It flagged B7H3, which later becomes a major target, and it noticed immune dynamics they would later study at single-cell resolution. Now their setup is more advanced. Jacob describes a system where he asks a natural language question, then spins up agents that do literature search, form a hypothesis, structure a bioinformatics analysis, execute it, and return a report with plots and even the Python code. One example was a scare around clonal hematopoiesis of indeterminate potential, CHIP, which can, if it goes wrong, lead to leukemia. They had a hint it might be happening in SID, potentially as a side effect of older chemo. Jacob asked their system to investigate. It cost about $20 in API calls, took about 30 minutes, and analyzed around 600,000 single cells from Sid's blood across multiple time points. He's explicit about the boundary. You don't trust it out of the box. It can make mistakes. The value is speed to competence, getting oriented fast enough to talk to specialists intelligently. In this case, they were able to put that risk to bed. That same AI-assisted competence shows up in the wildest part of the story, when they stop asking which drug to try and start asking what drug to build. They built a personalized mRNA cancer vaccine. Same basic idea as familiar vaccines, teach the immune system to recognize something foreign. But here, foreign is the mutations that distinguish Sid's cancer from his normal tissue. Sid was the first patient in an investigator-initiated trial. A lot of it is selection. You can encode only so much. Picking which antigens make the cut matters. Sid calls this more art than science today, but he's seen companies starting to use AI to do that selection automatically. That matters because if it stays manual, it will never scale. They went from project start to injection in 6 months, and they expect those timelines to come down. They also prepared more aggressive options, including TCR T-cell therapy and CAR T-cell therapy, described as the super killy insurance policy. Then Maximal Diagnostics throws them a curveball. It's the kind of detail that decides whether a nuclear bomb is even usable. They selected B7H3 as a CAR T target, but wanted to know where else it shows up in Sid's body. Genetic data helps, but nothing beats an actual scan. They found a scan in Beijing, and Sid traveled there. Good news, they didn't see any cancer. Bad news, his liver showed uptake 3.5 times higher than the 20 people scanned before him. That's the kind of information that makes a powerful therapy look like a way to lose your liver. So they went back to the partner making the therapy, Paul Robo, and asked if anything could be done. He'd invented a logic gate for CAR Ts, an AND gate, where 2 things have to be present before the therapy activates. They looked for something not present in the liver. FAP fit, based on earlier work showing low liver expression. So they designed it so both B7H3 and FAP have to be present. The nuclear bomb shouldn't go off in the liver. That's the feedback loop they keep emphasizing. Better diagnostics change the medicine design. And then they find a target that's almost too good. So good, it raises a new question. Why has nobody talked about it? Sid describes a gene target they call PENX3, showing up 10,000 times more in his cancer than in his healthy tissue. That's the kind of differential expression you dream about. They go searching for therapies and find basically nothing, barely a mention in the literature. That's strange, because most protein targets, people publish on them. Their hypothesis is why it's been missed. It's hydrophobic, and if you run typical tests in water, you might not find it. They only surfaced it by going back into the data and doing a painstaking analysis. This is where Sid gives AI a very human compliment. It has more patience than a person. It will trawl through terabytes and keep going until something pops. Underneath all of this is a more uncomfortable claim. Even if you have a good doctor, the system isn't tuned to your objective function. It's tuned to theirs. A line that sticks is about incentives. The doctors, Sid argues, want to minimize liability, which gets worse when a prescribed treatment causes severe side effects. As a patient facing an aggressive cancer, the objective can flip. He'd rather die from a treatment than from the cancer. That gap can mean people get too few medicines too late. AI, in their view, helps a patient show up differently. Not as someone demanding magic, but as someone able to discuss combinations, side effect overlap, organ-by-organ risk, and plausible reasoning even when randomized trials don't exist for every combination. And they keep pointing out the surprisingly affordable parts. Bulk RNA sequencing for $50. Whole genome sequencing starting at $500. A deep AI query for around $20. The bottlenecks they want to attack are bigger than one patient, especially clinical trials. Sid calls out slow FDA startup processes and points to Australia's notification approach as something that looks just as safe from the safety data he's seen. Through it all, the emotional core is simpler than the technology. When a radiology report said his lungs lit up like a Christmas tree, he got 15 minutes of hopelessness and prepared for the end. One doctor noticed the spread pattern didn't match osteosarcoma, guessed it wasn't cancer, and it turned out to be remnants of COVID. That's the point of this whole approach. More eyes, more hypotheses, faster iteration. If you want the full story, straight from Sijbrandij and Jacob Stern, listen to the OpenAI podcast episode, ChatGPT and Cancer, How a Tech Founder Rewrote His Treatment Plan. Then, ask yourself a hard question they keep daring people to ask. If medicine 30 years from now looks like this, who gets to live long enough to benefit from it?